All right, welcome back. We are going to continue our discussion of the molecular structure of DNA and RNA. This time we are going to look at both the structural features of a DNA strand. So for learning outcomes, describe the structural features of a DNA strand, but we are also going to look at the experiments that led to its discovery. So I would like you to be able to outline the key experiments that led to discovery of the DNA double helix. All right, so let's start off here with the structural features of a DNA strand. So we know that we have a polymer of nucleotides. These nucleotides are covalently linked together, and this linkage is called a phosphodiester bond. And when we look at the actual structure, we'll dig into this word a little bit, but in brief, there is a phosphate group attached via oxygens, which is called an ester bond, to the 5' or 3' carbons of the sugar. And so we have phosphodiester bond. Now, the fact that we have the phosphates connecting of 5' of one nucleotide to the 3' of another nucleotide, it means that the strand has directionality, meaning that if you look at one end of the strand, it looks physically different from the other end of the strand, based upon what part of the sugar molecule you're looking at. So it has directionality. And this directionality is usually stated as being 5' to 3' end, or the carbon number 5 to carbon number 3. And the only thing we mean by this is the direction you would want to read the DNA. Just as if we're looking at an individual sentence, we read from left to right. In English, in DNA, we read from the 5' end to the 3' end. Now, all of these sugars are orientated the same way on an individual strand, and the alternation of phosphate and sugar makes up the backbone of the nucleic acid strand. And this backbone looks the same for all pieces of DNA. So let's take an actual look here. You can see that we have a deoxyribose sugar right here. You can tell it's deoxyribose because if you look at number 2, there's no oxygen. So here on the 5' end, we have a phosphate group attached to that carbon. And when you look here, we have phosphate, covalent link to oxygen, covalent link to carbon. When you have things bonded to an oxygen, this is called an ester, and so it's a phosphate diester bond. Now, this is nucleotide number 1. If we go down to nucleotide number 2, we can see that its phosphate is linked up to carbon number 3 and to carbon number 5 of nucleotide 2. And so here, if we take a look at the overall pattern, we have a phosphate, carbon 5, carbon 3, 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 and so on and so forth. So we definitely have a directionality. So this is the 5' end, and this is the 3' end. Hanging off to the side here, we have the individual bases, and it can be any one of the four DNA, so thymine, adenine, cytosine, or guanine. And notice they all hang out here to one side. If you take a look at this side of the DNA, this backbone side, if you are approaching the strand from the left, it all looks the same regardless of what base pair is there. So, all we see as we walk around down this chain is a phosphate, sugar, phosphate, sugar, phosphate, sugar. So there are some proteins that are going to bond to DNA, and it's bonding to this type of pattern. There are going to be other things that interact with the DNA that recognize specific bases, and that is going to be binding in a slightly different way. So if we were to read this piece of DNA, we would read it as being TACG. All right, so let's dig into some of the experiments that led to discovery of a double helix before we actually get into the fine detail of the double helix itself. So, the paper that was published by Watson and Crick that described the structure of DNA was published in 1953. But this work was founded upon other people's work. And some key experiments that occurred here are some experiments by Linus Pauling, some experiments by Rosalind Franklin and Maurice Wilkins. They were working at the same university, in the same college. And then there's some work of Kargaff that had a key piece of information. So, Linus Pauling was a structural chemist, and he deduced the structure of the alpha helix. And this is the same alpha helix that you see in proteins, right? So he was able to deduce the structure of this based upon the molecules that were present, but also something called X-ray diffraction. So if you take a bunch of alpha helices and build them into a crystal and shoot X-rays through them, the repeating pattern in the crystal is going to cause the X-rays to kind of refract or bend in particular ways as it runs through these molecules. Based upon the pattern that comes out of that, you can deduce some key pieces of information, one of them being this helical type of content. So Linus Pauling had discovered this in the early 1950s. And consequently, after he had solved this structure here, he began to work on DNA. And the fact that Linus Pauling, who was famous for this, was now working on DNA, essentially was giving everyone else a strong push to finish their work before he got to it first. Now, we have Rosalind Franklin, and she was also an X-ray crystallographer. She was not really seeped heavily in chemistry or biology. She was really a physicist. And the things that she really could do well is fine-tune all of the instruments to get these extraordinarily sharp X-ray diffraction patterns. So here we have an X-ray source. It shoots out a beam of X-rays. They're all straight and parallel. They hit this crystal of DNA, and the fact that you have repeating patterns in the crystal cause X-rays to diffract in a particular pattern based upon what it's going through. Based upon this, you could tell a certain number of things. One is that there was a helix involved. Number two, you could deduce that there was more than one strand of DNA. And the last thing that you could really pull out of this is that there are 10 base pairs per complete turn. And we'll come back into this a little bit later. Now, you may have also heard about Rosalind Franklin from the standpoint of some controversy that occurred between her and Watson and Crick. And if you want to talk about this in class, we can dive into that. For here, I'm just gonna let it go. You might go watch the movie Double Helix. That kind of gives you a snapshot into some things that were happening there. All right. Another key experiment that is necessary is the work of Erwin Kargaff. And what he essentially did is this started taking DNA from different sources. And what he would do is essentially take that DNA from a particular source. He would get rid of all the proteins in that mixture by adding something called protease, which is an enzyme that cleaves proteins. So you would take a complex mixture of DNA and proteins, you'd get rid of all the proteins, then you dump in acid, and the acid would cause the individual bases to fall apart. You could then take that and separate them based upon size and chemical properties, and you could get an idea of how much A's there were relative to how much C, relative to how much G, relative to how much C. So if you did this for E. coli, you would notice that you have largely about 25% of all of them, right? And that doesn't tell you much. It just seems to enforce the idea that all of the bases occur equally. But if you start looking at different species, you'll start to see different patterns evolving. So if you look at Streptococcus pneumoniae, again, this is related to the paper that we're looking at, the pneumococcus that we're investigating there. Here, if you look at A's and T's, you've got about 30%, but G's and C's only have about 19%. Okay? This is different than we see in E. coli. What's interesting here is that T's and A's have the same percentage, and G's and C's have the same percentage, right? If you then take yeast and look at it, it is not exactly the same, but it's fairly similar. Here we have more like 32% A's and T's and 18% G's and C's. If we take a look at turtles, we get about 30%, or more like 28% here and 28% here, but then only 22% over here and over here. The important thing to pull out from this is, regardless of where you source the DNA, the percentage of A's always matches percentage of T, and percentage of G always matches percentage of C, right? So A's always equal T, and C's always equal G's. This observation was a crucial piece of information for Watson and Crick as they were starting to build their model and figuring out how the two pieces would fit together.